one of the things we really enjoy doing here is diving into the research you send our way. And today, we've got a really interesting study.
Yeah. What's it about?
It takes us down to Florida, to Biscayne National Park. A listener shared this, curious about, well, potential chemical presence in the water there.
Ah, Biscayne. Okay.
Yeah, and this study offers a kind of first look.
Right. So the specific paper we're unpacking is called Prevalence of Selected UV Filter Compounds in Biscayne National Park. It's by Landwehr and colleagues, published just this year, 2024, in Environmental Monitoring and Assessment.
And they frame it as a baseline, right? Like step one.
Exactly. That's key. It's a baseline assessment, a starting point for understanding what's actually in this pretty unique marine environment.
Okay, so our mission for this deep dive is basically to get a handle on what these scientists found. You know, what chemicals are there, what might it mean for the park. We want to pull out the main insights without getting totally bogged down in the super technical bits.
Sounds good. And the specific chemicals they were hunting for were 4 compounds: oxybenzone, dioxybenzone, benzophenone, and also benzocaine.
Okay, some of those sound familiar. Oxybenzone, that's in sunscreen, yeah.
That's the most common one people know, yes. A UV filter in lots of sunscreens and other products.
Right.
And benzocaine, that's more of a topical pain reliever you might find in some creams.
Right. So Biscayne National Park, why focus there? It seems like such a pristine place. Why worry about these chemicals?
Well, that's exactly why it's important. It's not just any park. It's actually the largest marine national park in the U.S. system.
Wow. Largest. Okay.
Yeah. And critically, it's right next door to Miami, a huge urban area.
Ah, the proximity factor.
Definitely. Plus, and this is really significant, the park protects a big chunk of the Florida Reef Tract. That's the 3rd largest barrier reef system in the world.
Okay, having a world-class reef system so close to a major city.
Yeah.
I can see the potential issue there.
Precisely. The worry is straightforward. You have lots of people swimming, boating, enjoying the park.
Mm-hmm.
Could they be introducing these UV filters mainly from sunscreen washing off?
And potentially harming the coral.
That's the concern, especially for the corals. And this study, it's actually the first time anyone's specifically measured these particular chemicals in Biscayne Bay itself, so it's filling a knowledge gap.
Okay, so we understand the why. How did they actually do it? How do you go about finding these potentially tiny amounts of chemicals in the bay?
Good question. They chose 5 specific spots inside the park for sampling. 4 were near the barrier islands, places called Boca Chica, Sands Key, Billy's Point, Caesar Creek.
Right, closer to land.
And the 5th one was out at Anniversary Reef. That's a really popular spot for visitors, snorkelers, divers. It's about 4 miles offshore.
Makes sense. Covering different types of areas, high-use reef and closer-in spots, did they take many samples?
They collected 35 water samples in total across those 5 sites, mostly during April of 2017 and 2018. Anniversary Reef had the most, 24 samples over those 2 periods.
Okay.
And importantly, they made sure to collect them during the daytime, when people were actually likely to be visiting and using the park.
Trying to catch it, yeah, when the input might be happening.
Exactly. Then, to figure out what was in the water, they used a pretty advanced technique. It's called liquid chromatography, coupled to high-resolution mass spectrometry.
Okay, that sounds fancy, like super-powered chemical detection.
That's a good way to put it. It basically separates everything in the water sample very finely, and then identifies and measures the exact amount of the specific chemicals they're looking for, even if it's a really tiny concentration.
Were there any, tricky parts to the sampling itself? Things they had to account for?
Yeah, that's a really smart question. Usually for this kind of work, scientists prefer amber glass bottles, but getting those out to some of the more remote spots, especially offshore.
Bit of a hassle, maybe heavy or breakable.
Right. So they wanted to use lighter, peaky plastic bottles. But first, they had to check if that would mess up the results.
So they tested the bottles.
They did. They compared PT versus glass using water spiked with a known amount of oxybenzone.
And did the plastic cause problems?
Interestingly, no. They found no statistically significant difference for oxybenzone between the PT and glass. So they felt confident using the PT bottles was okay for their study.
Good to know they checked that.
Yeah, and they also figured out their method detection limits, the MDLs. That's basically the smallest amount their machines could reliably detect. So they weren't just picking up random noise.
Right.
And they used field blanks, basically, pure water samples carried through the whole process, which showed no contamination. That helps confirm the samples themselves weren't getting contaminated during collection or handling.
Okay, sounds like they were careful. So the big question, what did they actually find? Let's start with oxybenzone. That's the one everyone talks about with corals.
Right. So oxybenzone It was detected in 26% of all the samples they took across the park.
Okay, about a quarter of them. How much did they find?
The highest level they measured was 31 nanograms per liter. And just for context, a nanogram is tiny, a billionth of a gram in a liter of water.
31.
In
Gel. Okay, incredibly small amounts. Did it vary much between the sites?
It did. Anniversary Reef, the busy reef spot, had it in 25% of samples, maxing out at 27 NGL. Billy's Point was similar at 25% detection, max 28 NGL.
Okay.
Boca Chica and Cedar Creek both had slightly higher detection rates, 33%, with maximums of 22 and 31 NGL respectively.
Yeah.
So Cedar Creek had the highest single reading.
Huh. And Sands Key.
Sands Key. Interestingly, in the one sample they analyzed from there, they found no detectable oxybenzone at all.
That is interesting. You might almost expect Anniversary Reef with all the people to be highest, but it wasn't the peak.
It is a bit counterintuitive. The researchers suggest that places like Sands Key and Anniversary Reef are in areas with more water movement tidal channels, open ocean.
So maybe things get diluted or flushed out faster.
That's the idea. More mixing, more dilution. But they're cautious. They say you'd really need more samples, especially from Sands Key, to be sure about that pattern.
Okay, so 31 NGL max, how does that compare? Like, is that a lot or a little compared to other places studied around the world?
That's the crucial context. The paper gives some comparisons. For instance, a lake in South Korea, max reported was only 1.2 NGL.
Wow, tiny.
But then you look at Trunk Bay in the U.S. Virgin Islands.
Mm.
They reported a max concentration of 1,395,000 NGL.
Whoa, that's orders of magnitude higher. A huge difference.
Massive difference. Other spots mentioned, like the Ionian Sea or a river in China, were generally much, much lower than Trunk Bay, too.
Mm.
So yeah, Biscayne's 31 NGL really does seem to be on the lower end of reported concentrations globally.
Okay, so relatively low levels in Biscayne compared to some hotspots. But even at 31 NGL, what's the thinking on whether that could affect the corals?
Yeah, this is where this discussion gets quite nuanced. Oxybenzone is definitely the most studied UV filter when it comes to coral health. And some research has suggested potential negative effects even at low concentrations, things like contributing to coral bleaching or harming coral larva development. The study mentions research pointing to bleaching effects from low sunscreen concentrations, for example.
Mm-hmm.
Another study found issues with coral larva development with an LC50, the lethal concentration for 50% of 139 micrograms per liter, and an EC20 affecting 20% at 6.5 micrograms per liter. There's even work suggesting oxybenzone might make corals more vulnerable to heat stress at levels like 60 NGL.
So their finding of 31 NGL, it's below that 60 NGL, but it's in that sort of low nanogram range where some studies have raised flags.
Exactly. It falls into that zone.
Yeah.
However, and this is a big, however, the research isn't all pointing in the same direction. There's conflicting data.
Okay.
Other studies have found that much, much higher concentrations are needed to cause significant harm. One study on a type of stony coral, for example, found an LC50 way up at 4.45 milligrams per liter, not micrograms or nanograms.
Milligrams, that's way higher.
Way higher. And a scientific review that looked across many studies calculated an overall EC50, the concentration affecting 50% for oxybenzone toxicity, to corals at 49 micrograms per liter. That's still much higher than the levels typically found around reefs, including the max level found in Biscayne.
So it sounds like the science isn't totally settled on exactly what concentration is safe or harmful.
You nailed it. That's a major challenge. The authors of this Biscayne paper point this out directly. There aren't standardized methods for testing coral toxicity for these chemicals.
Which makes it hard to compare studies or set clear environmental rules.
Precisely. Different methods, different coral species, different conditions. It leads to varying results.
Okay, so given all that complexity, what was this study's final assessment of the risk from oxybenzone in Biscayne National Park?
They calculated what's called a risk quotient. Basically, they compare the highest concentration they found at 31 NGL to that EC50 value from the review study, the 49 micrograms per liter. Doing that math gave them a very small number, a risk quotient of 6.3 times 10 to the minus 4.
Which means?
Based on that calculation, their conclusion is that, right now, the levels of oxybenzone they measured are not likely to pose a significant threat to the corals in Biscayne National Park.
Okay. That's somewhat reassuring, at least based on their findings and that risk assessment approach.
Yeah.
What about the other chemicals? Benzophenone was also on the list?
Yes, benzophenone.
Yeah.
And interestingly, they found that one more often than oxybenzone.
Oh, really?
Yeah, it was detected in 49% of the samples, almost half. The maximum concentration was higher, too, at 131 NGL.
Wow, okay. More frequent and higher levels. And wasn't Sands Key the place with no oxybenzone?
That's right. But its single sample did have benzophenone detected at 26 NGL. So 100% detection in that one sample.
So where's this benzophenone coming from? You said it's not really a U.S. sunscreen ingredient.
Correct. It's not an approved additive for sunscreens here. So its presence probably points to other sources.
Like what?
Well, the researchers suggest a few things. It could be forming from the breakdown of other UV filters in the environment, like octocrylene, which is used in sunscreens.
Ah, okay, a degradation product.
Possibly. Or it could be coming from other sources entirely. Benzophenone is used in things like cosmetics, some plastics to stop UV damage, even fragrances. It could also be getting into the bay via wastewater discharge, or maybe runoff from land.
So multiple potential pathways, not just people swimming with sunscreen. That makes its presence more complicated to interpret.
Definitely. It highlights that other human activities, beyond just recreation, might be contributing chemicals to the park's waters.
Oh, okay. And the last two, dioxybenzone and benzocaine, any sign of those?
Nope, neither of those were found above the detection limits in any of the 75 samples.
So good news on those fronts. Seems like they aren't current issues there.
Based on this snapshot, yes. It suggests they aren't significant pollutants of concern in the park right now.
Okay, so let's pull this together. We have oxybenzone present, but at levels deemed low risk by this study's assessment. We have more frequent benzophenone, likely from mixed sources, and nothing for the other two. What are the sort of broader implications here? Where does the research go next?
Well, one interesting point the researchers make is that even though oxybenzone levels were low, the fact that popular spots weren't necessarily the highest suggests water movement and dilution are really important factors, as we discussed.
Right. It's not just about where people are.
Exactly. But they do stress, again, needing more samples for a clearer picture. As for future work, they suggest a couple of interesting directions. One is to actually look for the breakdown products of oxybenzone.
Metabolites, like what happens after organisms process it.
Yeah, exactly. Since we know things can biodegrade oxybenzone, looking for those metabolites could give a fuller picture of its fate in the ecosystem.
That makes sense.
They also suggest testing surfaces within the park. Think algae, sediment, maybe even boat hulls or docks, to see if these UV filters are sticking to them, essentially being removed from the water column through adsorption.
So the water concentration is only part of the story. Where else might these chemicals be ending up?
Precisely. It's about understanding the whole system.
So overall, this study gives us a really valuable first baseline for Biscayne. The oxybenzone levels, while detectable, seem relatively low compared to some other global sites and aren't flagged as an immediate major threat by this assessment.
Correct. But the presence of benzophenone, possibly from non-sunscreen sources, is definitely a finding that warrants attention. It reminds us that our chemical footprint is complex.
It really does.
Yeah.
Thinking about these compounds, traveling from everyday products, maybe degrading from something else, ending up in a protected marine park like Biscayne, it makes you wonder, doesn't it? What other things, chemicals we don't even think about, might be finding their way into these supposedly pristine environments, even at these tiny, tiny concentrations? What are the subtle long-term effects we might not even be looking for yet? The study gives us a snapshot, a really important one, but it feels like the full story of how our activities chemically shape these natural places.
Well, that story is definitely still being written.